Hello everyone. In today's video, we are diving into an interesting topic in chemistry, the specificity of enzyme action, including stereospecificity. We'll understand why enzymes choose only certain substrates, how their active site work like perfectly molecule locks, and what makes them so selective in chemical reactions. So let's get started. Firstly, we will introduce to enzymes. Enzymes are biocatalysts with precise molecular architecture. The chemical reactivity depends on active site residue and electronic distribution, and the selectivity is essential for chemical transformation pathways. Now, what is the chemical nature of enzyme? Enzymes are basically proteins with catalytic amino acids like serine, histidine, and cysteine. Chemical behavior influenced by pKa, polarity, and steric factors. The catalytic efficiency explained via reaction coordinates diagram. Now, what are the active site chemistry? Spatial orientation of residue forms catalytic pockets, and the electronic stabilization of intermediates occur through hydrogen bonding. And the shape and electron density define specificity. Now, the concept of specificity. It is the specificity depends on geometric complementary. The electronic complementarity ensures correct reaction pathway and it determines which reactant undergoes transformation. Now, what are the types of specificity? These are the four types we can see. First is absolute, which has only one substrate fits. Second is group, in which substrates with similar functional groups. Third is bond, in which it recognizes specific chemical bonds. Fourth one is stereospecific. It distinguishes stereoisomers. So let's start with absolute specificity. For example, urease hydrolyzes only urea. The chemical reason behind that is planar resonance stabilized carbonyl fits perfectly in that, and it the attack occur at electronic deficient carbonyl carbon. Second one is group specificity. It acts on alcohols, amines, etc. For example, hexokinase, phosphorylates, glucose, and fructose. They depend on presence of hydroxyl groups forming hydrogen bonds. Third one is bond specificity. Lipase reacts with ester bond via nucleophilic attack. The mechanism involves serine, histidine, and asparagine catalytic triad. It recognizes polarized carbonyl carbon of ester. Now the chirality concept. What is chirality concept? So let's understand. Molecule with chiral center show stereoisomers. Enzymes bind one enantiomer in three-point attachment. For example, lactate dehydrogenase prefers L-lactate. Now what is transition state chemistry? It is simply when enzymes stabilize specific transition state and electronic interaction lowers activation energy. It determines stereospecific reaction rates. Now, the reaction mechanism. It involves nucleophilic or electrophilic reactions. Proper substrate orientation critical, and the specificity depends on orbital interaction. Now, what is the metabolic importance? The precise reaction prevent unwanted byproducts. And the stereospecificity ensures proper metabolic flow. Key in glycolysis. It is the key in glycolysis, TCA cycle, and amino acid metabolism. Now, the urea structure. We all know that the formula of urea is NH2CONH2. It is a resonance stabilizes planar structure which fits exactly into urea's active pocket. The glucose structure is, as we all know, that hexagonal pyranose ring with strong intermolecular hydrogen bonds, with C6OH bond phosphorylated by hexokinase. The ester functional group, which has a general formula RCOOR dash, carbonyl, in which the carbonyl carbon is electrophilic, and the lipase performs nucleophilic acyl substitution. Now, what is stereospecific enzymes? These are L-amino acid oxidase reacts only on L-amino acids. The active site geometry excludes B forms and it ensures correct protein metabolism. What are the industrial importance? So they used in asymmetric synthesis. Enzyme produced in enantiomerically pure compounds, and these are very crucial in pharmaceuticals. So let's study one case study, which is thalidomide, in which R enantiomer therapeutic and S enantiomer is harm. It shows need for stereospecific metabolic enzymes, and the enzyme pathway metabolize enantiomers differently. So if we summarize the whole topic, it will be like the enzyme specificity depends on geometry, electronics, and stereochemistry, and the key to chemical reaction pathways. And it is also essential in metabolism, catalysis, and drug design. Thank you very much.